Good morning, students. Today we'll start a new chapter. How do organisms reproduce? Now, in the chapter life process, we have read that every living being grow old and then they die. In fact, every living being remain alive for a certain period of time and then they die. So, new organisms have to be produced in that place, those who die. So the production of a new organism in the place of that organism, that is from the existing one of the same species, is called reproduction. Now this reproduction is essential for survival of the species on the earth. Now this process ensures the continuity of life on the earth. So, in the chapter heredity, we have read that the basic event of reproduction is the creation of the DNA copy. Now, this DNA copy has some importance. First one is the characteristics from the parents are transferred from the parents to the offspring during reproduction. Second importance of DNA copying is that some variations are produced in the offspring during the reproduction, which can cause the evolution. So now we will start about the reproduction. Now in this chapter there are we will learn about two types of reproduction. So first one is the asexual reproduction, second one is the sexual reproduction. So asexual reproduction where the production of the new organisms takes place by the help of single parent without the involvement of the sex cell. And sexual reproduction, the production of the new organism from the two parents by involving the two parents with the involvement of the sex cells, that is from their sex cells, by making use of their sex cells. So today we will learn about the asexual reproduction. Now in asexual reproduction there are some 7 types are there, 7 ways are there by which single parents or single organism give rise to their next generation. First one is the fission. So fission is the method by which one organism can divide or split into 2 or more organisms. So that is the unicellular one. So first one is the binary fission. In case of binary fission, that is the example is amoeba. Now when the amoeba cell has reached the maximum size of their growth, the nucleus lengthens and then they divide into 2 parts. Then the cytoplasm also divides and surrounds the nucleus, and this way the one amoeba, that is parent amoeba, divides into 2 daughter amoeba. Second one is the multiple fission. In multiple fission, this takes place when the is actually the 2 or more, that is the more amount of this splits are taking place. That is the parent organism splits into many new organisms at the same time. Sometimes a cyst or protective wall forms around the cell of single cell organisms like the Plasmodium. Now inside the cyst, the nucleus of the cell divides into many, and then they forms the daughter nuclei, many daughter nuclei. Now even the cytoplasm also divides and surround them, and a thin membrane also surrounds the whole thing. So this way inside the cyst many daughter nuclei are formed. Now when the favorable conditions comes, this cyst bursts open and then all the daughter cells are released and each form a new organism. Actually Plasmodium is the malarial parasite which causes the malaria. Next one is the fragmentation. So fragmentation is the breaking up of simple multicellular organisms into 2 or more parts. Eventually each part can grow into a new individual. like in case of Spirogyra. But this is not true in case of all the multicellular organisms, because they cannot divide cell by cell. Next is the regeneration. So regeneration is the process in which this actually many fully differentiated organisms can give rise to many organisms from their own body parts. In fact, the individual is somehow cut or broken into many pieces, and these pieces can grow into a new organisms, and this process is called regeneration. Regeneration is carried out by the specialized cells, and these cells proliferate and make large amount of cells. That is from this mass of cells, the different cell grow and become some different cell types of the tissues. This is carried out by the Hydra or Planaria. Next one is the budding. Now in budding process, in case of Hydra, the body of Hydra, from the body of Hydra, a bud that is an outgrowth grows, that is develops, and because due to the because of the division of specific cells and at the specific site. Now this then develops, this bird then develops into tiny individual, and when they become totally mature, they detach from the parent body and grow as an independent individual, like in case of Hydra. Next one is the vegetative propagation. So in some plants, the plants can be grown from the vegetative parts, that is the roots, stem and the leaves. Like in case of stem, we know that potato is a underground stem. If we put the potato with the bud in the soil, if it gets a favorable condition, it will grow into a new potato plant. Same way, from the roots of the dahlia can be grown. Next, last one is the leaves. In case of Bryophyllum leaves, we see the margin are notched one. So from the notches of the Bryophyllum leaves, the buds grow, and if that buds fall on the ground and gets the favorable conditions from that bud, it will grow into a new Bryophyllum plant. There are other artificial propagation is also there, that is by cutting method, layering method and the grafting method. By the cutting method, we can grow the rose plants. From the layering, we can grow the jasmine plants. But these are all artificial methods by man-made one. And from the grafting, that is grafting is the method by which two different stems are joined together so that they can grow as a single plant, and this way you can grow the apple. Actually, the advantages of this vegetative propagation is that the first one is the plants that is grown by this method can bear the flowers and the fruits are more easier or earlier than that is grown by the seeds. Second one is the plants which have lost the capacity of seeds for producing seeds. That plant also can be grown through this vegetative propagation method. Third one is these plants are genetically similar to that of the parent plants, having all its characteristics. Next one is the tissue culture. So in tissue culture, that is done by taking the small tissue, removing the tissue or separating the cell from the plant, from the growing tip of the plant. Then this cell are transferred to the medium where they can grow, that is where they can divide and multiple, and then it can become say mass of cell. Then this mass of cells are again transferred to the medium containing the hormones for growth and differentiation. Then the plantlets are placed into the soil from where they can grow into a mature plants. So this tissue culture method is done in the ornamental plants. Last one is the spore formation. So spore formation is the method actually. This is seen in case of some fungi like Rhizopus or Mucor. Then we can see it in the bacteria or some non-flowering plants like the fern or mosses. So in spore formation, the parent plants produces hundreds of microscopic reproductive units called the spores. When the spore case bursts open, then these spores are spread into the air, and when these airborne spores fall on the food and gets a favorable condition, they germinate to produce the new plant. Especially we see this in the braid, that is the common braid mold is the Rhizopus, where we see the fine thread like projections called the hyphae. Then thin stems are there. Over it the knob like structure is there, which is called sporangia. Inside the sporangia, spores are there, minute hundreds of spores are there in the spore case. When this spore case bursts, these spores fall on the food, and when they, especially on the, that is the moist bread, when this moist bread, they grow into the new Rhizoplus plant. So this way the asexual type is finished. Next we will start with the sexual reproduction.